Good morning, dear students. So today we are going to discuss digestion in the small and large intestine and absorption. So we should continue discussion digestive system physiology, and we'll complete this discussion today during the lecture. As you remember, the previous lecture we have discussed the general principles of GIT regulation, the basic processes and stages of digestion functions of digestive system. As well, we have discussed the main processes of digestion in the oral cavity, the mechanisms of regulation of saliva secretion, the motor activity, processes of mastication and deglutition, and also the main characteristics of digestion in the stomach, secretion, and the properties of gastric juice and mechanisms of its regulation, motility of the stomach, absorption in the stomach, and the mechanisms of regulation of stomach's secretion and motility. So today we are going to continue to discuss digestion in the GIT. And we will discuss the digestion in the small intestine, in the large intestine, and absorption. So, the small intestine is a portion of the gastrointestinal tract between the pyloric sphincter of the stomach and the ileocecal valve opening into a large intestine. Its length approximately 3 meters. The first 20 to 30 centimeters from the pyloric sphincter is the duodenum. The next 2/5 is the jejunum, and the last 3/5 is the ileum, which empties into a large intestine through the ileocecal valve. It receives chyme from the stomach. It receives pancreatic juice from pancreas, and it also receives bile from the gallbladder. So now let's discuss pancreatic secretion. The pancreas lies parallel to and beneath the stomach. It is a large compound gland with an internal structure similar to that of the salivary glands. It lies across the abdominal wall. The portion of the pancreas that secretes pancreatic juice is a compound alveolar gland resembling the salivary glands, and granules containing the digestive enzymes, so-called zymogen granules, are formed in the acinar cells and discharged by exocytosis from apexes of the acinar cells into the lumen of the pancreatic duct. The small ducts will drain into the common bile duct to form the ampulla of the duodenal papilla, which it is encircled by the sphincter of Oddi. The pancreatic juice, exocrine secretion, as I told already to you, is about 1.5 to 2.5 liters per day. Its pH ranges in between 7.5 to 8.8, and the cells it contains water 96 to 98%, and organic and inorganic compounds. Organic and inorganic compounds. The inorganic compounds are anions and cations, which you may see here, and the most important inorganic compound of pancreatic juice are bicarbonates. The bicarbonate ions are produced by the cells lining ducts, which also produce water. The bicarbonate, as you know, help along with bile and intestinal juice to neutralize gastric acid. Organic compounds of pancreatic juice are enzymes. The pancreatic enzymes are divided into enzymes that are so-called carbohydrate splitting enzymes, or amylolytic enzymes, alpha amylase, which splits starch to maltose and maltotreose and alpha limit dextrins, maltase, lactase. Then proteolytic enzymes or proteases. The proteolytic enzymes, as I told already, are secreted in inactive forms, zymogens, to protect pancreatic tissues from digestion. The proteolytic enzymes are following: chymotrypsin and trypsin, which are secreted in inactive form, trypsinogen and chymotrypsinogen. A trypsinogen activated by enterokinase of intestine, then it becomes active trypsin, and then trypsin autocatalytically will activate chymotrypsinogen, its conversion to chymotrypsin, and also procarboxypeptidases conversion to carboxypeptidases. Trypsin and chymotrypsin split polypeptides to peptides, carboxypeptidase and elastase also, and carboxypeptidase splits amino acids from carboxyl end of peptide chains. Then fat splitting enzymes or lipase. Pancreatic lipase splits triglycerides to monoglycerides and free fatty acids. Nucleases. The nucleases split nucleic acids to nucleotides. And phospholipase split phospholipids. Here on that slide, you may see demonstration of different types of functions of different types of pancreatic enzymes and their role in the digestion of complex organic molecules. The action of amylase on the starch is shown here, as you may see. The action of pancreatic lipase on fats splitting, and the action of proteolytic enzymes, proteases, on proteins splitting. And there are also the factors, neurogenic and humoral factors, that will increase, that will influence on the secretion of these enzymes by acinar cells of exocrine portion of pancreas, because today we are discussing only exocrine digestive function of pancreas, which is based on the secretion of pancreatic juice. There are four basic stimuli causing pancreatic secretion, which are acetylcholine released by vagus nerve postganglionic fibers, gastrin, which, as you know already, secreted by stomach's enteral mucosa, enteroendocrine cells, gastrointestinal hormone, one of famous gastrointestinal hormones. Then CCK, cholecystokinin, pancreosamine, and secretin, as well as the autonomic nervous system fibers. The various stimuli multiply or potentiate each other. Now we should discuss the phases of pancreatic juice secretion. Secretion of pancreatic juice is under two types of control. There are nervous and humoral control. The humoral mechanism is more important. So the nervous control of pancreatic juice based on the stimulation of the parasympathetic nerves, stimulation of vagus nerve. It can be divided into three phases. Cephalic phase. Here you may see the simple scheme of cephalic phase of pancreatic juice secretion, which is based on conditioned and unconditioned stimulation. As you remember, thought of food, sight of food, smell of food, even sounds of its preparation may initiate secretion of pancreatic juice as well as secretion of gastric juice and saliva. And also, as you understand, stimulation of taste receptors in the oral cavity, which is conditioned, unconditioned. So unconditioned and conditioned phases of pancreatic juice secretion will initiate secretion of pancreatic juice, small volume of pancreatic juice rich in enzymes, and also release a small quantity of gastrin from antrum, which may stimulate pancreas. So this cephalic and gastric phase of pancreatic juice secretion mostly depends upon the vagal reflexes. So it is realized through the vagal nerve endings in the pancreas, and it gives around 20% of pancreatic secretion. The nervous control also includes intestinal phase. The intestinal phase, which is based on the distension of the initial portion of the small intestine, duodenum by chyme entering the duodenum following gastric emptying. It also initiates the vagal reflexes and realized through the vagus nerve stimulation, and this stimulation also add into pancreatic secretion acetylcholine released by postganglionic parasympathetic stimulates DAT and acinar cells, and it is also important stimulus. the humoral control over pancreatic secretion. The humoral control involves two phases, gastric phase and the intestinal phase. The intestinal phase is the major control of pancreatic secretion. So the gastric phase of pancreatic secretion begins from the distension of the duodenum and the protein products. Distension of the stomach and protein products in the stomach, the products of proteins digestion in the stomach. It will cause release gastrin, which may stimulate the pancreas through the blood. The same time, because of that, the gut epithelium secretes watery juice rich in bicarbonates. Intestinal phase. The intestinal phase is based on the stimulation of chemoreceptors in the mucosa of the duodenum and jejunum by acid chyme, which entering here from the stomach, and by products of digestion, as for example triglycerides, fatty acid, peptides, and amino acids. These stimuli will cause secretion of secretin gastrointestinal hormone by the duodenal mucosa S cells, and secretion of CCK, cholecystokinin, pancreozymin by duodenal mucosa I cells. After acid chyme and the chyme which contain the products of digestion, fatty acids, amino acids, enters the small intestine, the pancreatic secretion becomes profuse, mainly in response to the hormone secretin that stimulates that epithelium to secrete large quantities of watery juice rich in bicarbonate, and fats and amino acids will cause a release of CCK that passes by way of the blood to the pancreas and cause secretion of large quantities of enzymes by acinar cells. And effect, as you see, similar to the effect of vagus nerve stimulation and also gastrin, but much pronounced. So one more time, should repeat you that this phase is the major control over pancreatic secretion, Pancreatic secretion stimulated by substances which I listed already above, which are acetylcholine, gastrin, CCK, and secretin, and it is inhibited by increased tone of sympathetic nervous system, pain, fear, and anger. It is inhibited by glucagon secretion, by somatostatin secretion from cells of pancreatic islets and intestinal mucosa. And there are also some factors, as for example vasopressin, substance P, GIP, which also can inhibit secretion of pancreatic juice. So here on that slide, you may see by the these blue arrows, the effects of parasympathetic nervous system, the effects of vagus nerves through the vagal reflexes on secretion of pancreatic enzymes. Strictly speaking, the vagus nerve initiates secretion of pancreatic juice, while humoral factors, the hormones, gastrointestinal hormones, mostly these hormones are secretin and CCK and some other also, but these are much more powerful and important, will stimulate profuse secretion over pancreatic juice. Now we should continue and discuss the functions of the liver in the process of digestion in the small intestine. As you know, the liver is the largest internal organ which lies immediately beneath the diaphragm in the abdominal cavity. The gallbladder attached to the internal surface of the liver, and the liver receives venous blood from the intestine, which passes through the portal system. The functions of the liver are following. The first function is based on removal of drugs, hormones, the detoxification of drugs, hormones, waste products of metabolism, and other chemicals. That's why this function called detoxification of blood. Liver also the central organ of proteins, fats, and carbohydrates metabolism. So, as you know, its participation in carbohydrates metabolism is based on maintenance normal blood glucose level. It converts glucose to glycogen and glycogen to glucose. Converts amino acids, lactic acid, fructose, and galactose to glucose, and converts glucose to triglycerides. So, the processes of glycogenolysis, glycogenesis, and gluconeogenesis takes place here. The liver also the central organ of proteins and fats metabolism. So it, as you know, it is responsible for proteins synthesis. For example, albumins, plasma transport proteins, plasma clotting factors, prothrombin, fibrinogen are synthesized here in the liver. It also removes NH2 from amino acids, which can then be used to form ATP. It converts toxic NH3 to the less toxic urea for excretion in urine. It stores triglycerides. Synthesizes cholesterol, which is used to make bile salts and also is released into the bloodstream. It synthesizes lipoproteins which transport fatty acids, triglycerides, and cholesterol. One of the functions of the liver also storage function. So it stores glycogen, fats, vitamin A, B12, vitamins D, E, and K, copper and iron. The endocrine function of the liver is based on secretion of insulin-like growth factor 1, EGF1, well known to you as somatomedin C, in response to growth hormone. It also forms T3 from T4, and it secretes angiotensinogen. But the most important function of the liver is its digestive function. The digestive function of the liver is based on secretion of bile. It secretes bile rich in bicarbonates, which helps neutralize acid in the duodenum. It also synthesizes bile salts from cholesterol. These, as you know, aid absorption of fats, cholesterol, phospholipids, and lipoproteins from the intestine. It synthesizes bile pigments from hem of hemoglobin, and it excretes plasma cholesterol and lecithin. Here on that slide, you may see the composition of bile. The bile secretion stays around 500 to 1000 milliliters per day. It composed of water, organic and inorganic substances. The water inorganic substances are electrolytes, anions, and cations, and the most important substances are bile salts, bile pigment bilirubin, phospholipids, lecithin, cholesterol. The bile pigment bilirubin, derivative of the hem group from the hemoglobin of all red blood cells. Bilirubin conjugated with glucuronic acid and can be secreted into the bile. By the way, it gives its yellow color and enters the intestine. Then it is converted by bacteria into urobilinogen, which is responsible for the color of the feces and urine. Bile salts are derivatives of cholesterol conjugated with glycolic acid and taurocolic acid and cations. These bile salts form aggregates known as micelles. Lipids in the small intestine, lecithin, cholesterol, and other, enter these micelles and involved in a process of digestion and absorption of fat. Here on that slide you may see the several functions of bile, which we have discussed already earlier. Emulsification of fats, absorption of fats, the formation of micelles, activation of pancreatic and intestinal enzymes and stimulation of hepatic and pancreatic secretion, neutralization of acid chyme, and bactericidal effect. Bile is discharged into hepatic ducts and in the gallbladder for storage. Here it will be concentrated because of water and salts reabsorption. It becomes concentrated 10 times more, and contraction of smooth muscles in the walls expels bile to the duodenum from the common bile duct and through the sphincter of Oddi. In intestine, bile acts on fat through its breakdown into smaller globules. When duodenum is empty of food, the sphincter of Oddi closed and bile stored in the gallbladder. During a meal, it is discharged into the duodenum. So, as you remember, the main digestive function of bile in intestine, it is that it breaks fat in food into small globules with negative charge on their surface and simplifies the process of fats digestion. As I told already, bile secreted continuously but increases during a meal. The nervous control ensures the contraction of gallbladder walls in response to vagal stimulation. And here you may see cephalic and gastric phase of reflex vagal stimulation of this process, bile expulsion of bile from the gallbladder. The humoral control is based on release of secretin and CCK from the duodenum. As you may see here on that scheme, there are also principally the same scheme of regulation of bile expulsion from the gallbladder. And here you may see nervous and humoral factors which will influence this expulsion. The nervous control of bile expulsion is based on stimulation of parasympathetic nerve. There are two phases of reflex control, which are cephalic phase, and cephalic is usually conditioned and unconditioned, and gastric phase. The stimulus are in cephalic phase, taste of food, it is unconditioned, and conditioned stimuli are sight of food, smell of food, thought of food, and even sounds of its preparation, as I told you earlier. Altogether, taste of food, sight, smell, and thought of food will stimulate the parasympathetic nerve, vagus nerve, and simultaneously to that, the distension of the stomach by the food entering the stomach also stimulates the vagus nerve afferents, and altogether it leads to contraction of gallbladder, relaxation of sphincter of Oddi, and bile entering the duodenum. As well, it also stimulates the bile production by the liver. Humoral or humoral regulation, humoral factor. It is based on products of fat digestion, fatty acids, proteins digestion, amino acids in the chyme entering the duodenum and stimulating the duodenal mucosa to secrete cholecystokinin into blood, and acid chyme entering the duodenum stimulates secretion of secretin. Secretin by S cells, as you remember, and CCK by I cells, enteroendocrine cells over GIT in the duodenal mucosa. These enteroendocrine hormones will act via the blood, and the result of this stimulation is contraction of gallbladder and bile expulsion, and bile duct epithelium secretes water and bicarbonates rich fluid, mostly potentiated by CCK. So the result of this humoral control, which is the major phase in the regulation of bile expulsion, which controls the contraction of the gallbladder, is contraction of gallbladder, expulsion of phase, expulsion of bile into the duodenum. During and just after meals, bile salts are absorbed in the distal ileum and carried via the portal blood to the liver, where they stimulate bile salts secretion by inhibit bile salts synthesis. Between meals, since release of bile to the duodenum is low, this is a low concentration of bile salts in the portal blood, producing inhibition of bile salt secretion and stimulation of bile salt synthesis. No fibers containing vasoactive intestinal peptide, the abbreviature VIP, and sympathetic nerves in the wall of the gallbladder inhibits contraction of the gallbladder, so inhibits expulsion of bile. So as you see, there are also two factors, nervous and humoral factors, which influence not only secretion of bile by liver, but also expulsion of bile from the gallbladder. And here you may see that the role of intestinal humoral phase is much more important in compare to the to its role in, for example, gastric juice secretion. Here you may see again the different factors, neural and humoral factors, which will influence on secretion of pancreatic juice by exocrine portion of pancreas, by acinar cells, and production and expulsion, cholerasis and cholekinesis, production and expulsion of bile from the gallbladder. By different colors, it is clear that there are vagus nerve stimulation, then the role of different gastrointestinal hormones as cholecystokinin, GIP, secretin, yeah, gastrin, in production of these types of secretion. I mean gastric juice secretion, pancreatic juice secretion into the duodenum, and bile secretion and expulsion into the duodenum. Now we should discuss the small intestine. The general aspects, will just briefly repeat some general aspects from morphological aspects. As you remember, the small intestine is the long muscular tube, approximately 275 centimeters long in life and 70 centimeters after death. It receives chyme in small quantities from the stomach. It receives pancreatic juice from the pancreas, as well bile from the gallbladder. Now briefly about morphological organization. The wall of the intestinal wall has 4 layers. The outer layer called serosa or peritoneum with vessels and nerves, yeah, serosa or peritoneum. Second layer called muscularis, which consists of 2 layers of muscles, longitudinal muscles and circular muscles. Between the muscular layers lies the mesenteric or Auerbach's nerve plexus, through which peristaltic movements are controlled. We discussed it at the first lecture. Here it is given by this yellow color, yeah, this yellow color threads. Third layer called submucosa with dense fibrous tissue in which lie blood vessels, lymphatic vessels, and submucous Meissner's nerve plexus, which coordinates motor and secretory activity of mucosa. And the last one called mucosa or lining. Mucosa has microscopic folds which increase the surface area. These folds called villi, and foldings of the apical membrane of epithelial cells called microvillis. This microvilli can only be seen in an electron microscope. Villis can be seen in light microscope. These microvillis produce so-called brush border. Now, secretion in the small intestine. The daily secretion in the small intestine ranges in between 2 to 2.5 liters per day. pH is alkaline. The intestinal secretion depends upon secretion of so-called duodenal only in the located only in the duodenum, Brunner's, mucus glands, and secretion of intestinal glands called Libercune glands or crypts of Libercune. The Brunner's glands, located in the first few centimeters of the duodenum, between the pylorus and the papilla of water, where the pancreatic juice and bile empty into the duodenum. These glands are responsible. These glands will secrete the mucus in response to tactile stimuli of the mucosa, vagal stimulation, and gastrointestinal hormones, especially secretin. The function of the mucus secreted by the Brunner's gland is to protect the duodenal wall from distension, from the digestion by acid gastric juice. The Brunner's glands are inhibited by sympathetic nervous system stimulation. Secretion over crypts of Libercune or small pits. It is almost pure extracellular fluid. It has a slightly alkaline pH. It is secretion of mucus and extracellular fluid. The epithelial cells of the mucosa, especially those covering the villi, do contain digestive enzymes. So, there are two types of secretory cells in the crypts of Libercune: the goblet cells, which secrete its secretion in response to tactile stimuli throughout the intestine, and enterocytes, which secrete the hormones, which secrete enzymes. Intestinal juice composition. The brush border enzymes are classified according to the substances digested, as usually, are classified as protein splitting enzymes, carbohydrate splitting enzymes, fat splitting enzymes, nucleotide splitting enzymes. Generally, there are more than 20 types of different classes. Enzymes are secreted by the intestinal glands and will act as a brush border enzymes. The brush border enzymes will complete the process of organic molecules digestion and which finally will lead to absorption of the products of digestion from the lumen through the apical membrane of enterocytes. The intestinal juice also contains mucus, as I told you, water, epithelial cells, and non-organic substances. As I told already, the secretion of juice and regulation of juice secretion depends upon the local nervous reflexes in response to tactile stimuli of the mucosa, and it also depends upon the hormones, gastrointestinal hormones, secretin and CCK. But the first type of control local nervous reflex control plays a dominant role in the secretion of intestinal juice. The types of digestion in the small intestine are cavital, in the cavities, in the gut lumen, due to enzymes of pancreatic juice, due to bile action, and membrane digestion, membranous digestion, due to the brush water enzymes on membranes of the enterocytes. The movements of the small intestine are, as elsewhere in the gastrointestinal tract, can be divided into mixing contractions and propulsive contractions. This separation is a bit artificial because essentially all movements of the small intestine cause at least some degree of both mixing and propulsion. But usual classification of movement of the small intestine, motility of the small intestine, are following. First one, mixing contraction, called rhythmical segmentation. Rhythmical segmentation. When a portion of the small intestine becomes distended with chyme, the stretch of the intestinal wall elicits localized concentric contractions spaced at intervals along the intestine. As one set of segmentation contraction relaxes, a new set begins. It promotes progressive mixing of the solid food particles with the secretions of the small intestine. This type of movement is myogenic. It is a property of the smooth muscle cells. It does not depend on a nervous mechanism. The frequency of segmentation contractions is determined by the frequency of the slow waves. We have discussed these slow waves and peristalsis at the previous lecture. These slow waves, as you remember, is the basic electrical rhythm. The frequency of the segmentation contractions is about 8 to 12 per minute. Next type of small intestine motility is peristalsis, the peristalsis in the small intestine. Chyme is propelled through the small intestine by peristaltic waves which move at the velocity of about 0.5 to 2 centimeters per second. The net movement of the chyme along the small intestine averages only 1 centimeter per minute. Normally, 3 to 5 hours are required for passage of chyme from the pylorus to the ileocecal valve, and this type of movement is neurogenic. It is carried out through a local reflexes mediated through intrinsic nerve plexuses within the walls of the tube. The peristalsis is controlled by the chyme entering into the duodenum. The first factor is entering of chyme into the duodenum. And also gastroenteric reflex, which is initiated by the distension of the stomach and conducted through the myenteric plexus down from the stomach along the wall of the intestine. So reflex control, nervous control, mostly due to the myenteric plexus functions. And as usually, humoral control, gastrin, CCK, insulin, serotonin increase motility of the small intestine. On the other hand, secretin, glucagon inhibit motility of the small intestine. Motility I mean peristalsis, yeah? And the last one, the last type of motility of the small intestine, called movements of Willis or villus pump. Villus pump. These movements are not seen here, but these movements caused by the muscularic mucosa and the muscle fibers of the villi, initiated by the local nervous reflexes in the submucosal plexus that occurs in response to chyme. So as you see, the role of Auerbach's and Meissner's plexuses is very important in the processes of secretion and motility over the small intestine, as well as the role of vagal reflexes and the role of gastrointestinal hormones secreted by duodenal mucosa and which act via the blood. The large intestine. So, chyme from ileum, as you remember, from ileum, passes into the cecum, which is a blind ending pouch at the beginning of the large intestine or colon. Then it passes through the ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and anal canal. And feces is excreted through the anus. The mucosa of the large intestine contains many lymph nodules and is covered by columnar epithelial cells and goblet cells. As you may see here, there are no villi in the large intestine. It is flat. The epithelium forms crypts of Libercune, and the outer surface forms pouches or haustras. Principally, the functions of the colon are following: absorption of water and electrolytes from the chyme, the proximal half of the colon, and storage of fecal matter until it can be expelled in the distal half. The movements are similar to those of the small intestine we discussed a bit earlier, and can be divided into mixing movements, and propulsive movements. The mixing movements, also called haustration, you can see it here on the slide. Combined contraction of the circular and longitudinal smooth muscle contraction cause the unstimulated portion of the large intestine to bulge outward into bag-like sacs called haustrations. After a few minutes, new haustral contractions occur in other area nearby, and the fecal material exposed to the surface of large intestine and fluids and substances are absorbed. The second one, called propulsive movements or mass movements or mass peristalsis, which occurs a few times each day. So look here one more time. Segmentation, long-lasting myogenic contraction, reins of a circular muscle which divide colon into deep pockets, haustras, as I told already. As you know, this segmentation contraction means the contents of the colon and promotes absorption of water and salts, and the peristaltic waves will propel peristaltic waves, as you see here, will propel their contents towards the rectum. And sometimes, as you may see, the weak antiperistalsis is seen, antiperistalsis in back direction. And next one, mass peristalsis or mass movement. Simultaneous, single, powerful, long-lasting peristaltic contraction of circular smooth muscle that moves feces towards the rectum, and the rectal distension initiates the defecation reflex. Defecation or emptying is a complex reflex act. The passage of feces into the rectum distends walls and initiates reflex, which is prolonged by the passage of feces through anal canal. The desire or the urge to defecate reaches consciousness at rectal pressure is about 18 millimeters of mercury. When this pressure reaches 55 millimeters of mercury, the external as well as internal sphincters relax and the content of the rectum are expelled. The outgoing nerve impulses from mechano and baroreceptors of rectum are sent to the spinal cord S2 to S4, to the center of defecation. From that, ingoing motor fibers carry impulses along pelvic and pudendal nerves, two internal and external sphincters, respectively. It relaxes sphincters and increases motility of sigmoid colon and rectum. This is assisted by the increased intra-abdominal pressure by contraction of diaphragm and abdominal muscles. In humans, the sympathetic nerve supply contracts sphincters and inhibits motility of rectum, while parasympathetic nerves relax sphincters and increases motility, by that facilitating evacuation, defecation. And the last thing we should discuss today is absorption in GIT. As you remember, there are two most important major functions of the gastrointestinal tract are secretion, motility, and absorption. As you know, absorption is the process of moving nutrients from the gastrointestinal tract into the bloodstream. Majority of absorption takes place in the small intestine. The nutrients, the products of digestion, are absorbed via several types of transport, which you know very well. These types of transport are passive transport and active transport. The passive diffusion takes place when nutrients move from high concentration to low concentration and no energy is required. Facilitated diffusion happens when nutrients move from high concentration to low concentration with the help of a carrier proteins and also no energy is required. So these are types of subtypes of passive diffusion, passive transport, passive facilitated diffusion. There is also osmosis, as you remember, due to which the water is absorbed. So osmosis, passive and facilitated diffusion. And active transport. The active transport is when nutrients move from low concentration to high concentration with the help of a carrier protein and energy is required. And one more type, endocytosis, when cell forms vesicles to surround and engulf nutrients. So here on that slide you may see the different types of transport by which different types of nutrients, the product of carbohydrates digestion, proteins digestion, and fats digestion are absorbed from the gut lumen through the apical membrane of enterocytes. As we said already, most absorption occurs in the small intestine. Around 90% of substances are absorbed there. Nutrients are trapped in folds of the intestinal wall and absorbed through the microvilli. Each villus contains blood vessels and lymph vessels which transport the nutrients. Now let's discuss the absorption of products separately, the products of carbohydrates, proteins, and fats digestion. So digestion of food continues as it is absorbed through the brush border and cytosol of the endothelial cells of the small intestine. Carbohydrates. The digestion of starch, the long polysaccharide of glucose, begins, as you remember, in the mouth with the action of salivary amylase, which splits starch into maltose, 2 glucose units, maltotriose, and alpha-limit dextrins. But food stays in the mouth not enough for sufficient digestion, and the digestive action of salivary amylase limits when bolus mixes with gastric juice because of low pH of gastric juice. Then the digestion of starch occurs in the duodenum by the action of pancreatic amylase. It cleaves starch to produce the disaccharides and trisaccharides and oligosaccharides, sucrose, lactose, maltose, maltotriose, yeah? Maltose and maltotriose and oligosaccharides are hydrolyzed to their glucose, galactose, fructose to their monosaccharides by brush border enzymes located on the microvilli of the epithelial cells in the small intestine. Monosaccharides are then moved across the epithelial cells membrane by secondary active transport and facilitated diffusion. The secondary active is co-transport of glucose with the sodium. From cytosol, the monosaccharides pass by diffusion or facilitated diffusion through the basolateral membrane into the capillaries within the intestinal villi. Proteins absorption. The proteins digestion begins in the stomach, as you remember, with the action of pepsins. But the complete digestion of proteins occurs in the small intestine, in the duodenum and jejunum, due to the action of pancreatic juice proteolytic enzymes or proteases, as trypsin, chymotrypsin, elastase, carboxypeptidase, and then brush border enzyme aminopeptidase. As a result, polypeptide chains are digested into free amino acids and dipeptides and tripeptides. They are absorbed by secondary active transport using sodium co-transport system into the duodenal epithelial cells and secreted into the blood capillaries. The dipeptides and tripeptides may enter the epithelial cells by a different carrier systems, but they are digested within these cells into amino acids, which are secreted into the blood that carry them to the hepatic portal vein. And finally, the fats digestion. Fats or lipids absorption. The mixed micelles of bile salts, lecithin, and cholesterol are secreted into the duodenum and emulsificate a large fat droplets into much smaller and usable for digestion, for contact with enzymes, lipolytic enzymes. This increases the surface area for pancreatic lipase action, and pancreatic lipase digests fat triglycerides by cleaving off the first and third fatty acids. These produce free fatty acids and monoglycerides, which move then into the mixed micelles of bile salts, lecithin, and cholesterol. Fatty acids and monoglycerides from the micelles within the small intestine are absorbed by epithelial cells, brush border, and converted intracellularly into triglycerides. It is called resynthesis of triglycerides takes place. They are then combined with protein to form chylomicrons, which enter the lymphatic vessels, lacteals of the villi. These lymphatic vessels transport the chylomicrons by exocytosis to the thoracic duct, which empties them into the venous blood. So as you see, the process of fats absorption is a much more complicated and staged process. Water is absorbed by osmosis, as I told already, secondary to sodium ions absorption. Sodium, calcium, magnesium are absorbed by active transport. Potassium and chloride by passive transport. Vitamins are absorbed by passive transport. After absorption, the nutrients are transported in blood through the mesenteric veins to liver, then to hepatic veins, by hepatic veins to the inferior vena cava, and then to systemic circulation. And also the nutrients are transported by the lymph from lymphatic vessels to thoracic duct and to systemic circulation. So today we have discussed the last topic over physiology in gastrointestinal tract. Our lecture is over. Goodbye. Спасибо.